to introduce the Nature method of this semester. This paper is published on 2019 in Nature method. The title is Joint Profiling of DNA Methylation and Chromatin Architectures in Single Cells. This paper has to combine two important technologies, including the DNA methylation and also analyze the chromatin architecture in single cells. So my talk will be focused on three parts. The first is the DNA methylation. How do we detect the DNA methylation and why the DNA methylation in cells is important? And the second is that the chromatin architectures is hard to study. Therefore, the scientists developed a new technology which named Hi-C. So I will introduce what is the Hi-C technology. Moreover, the author also want to explore these two technologies combined in single cells. So we can see that first, the DNA methylation, which defined as the methylation at the fifth position in the cytosine, which was named as the 5-methyl cytosine. This cytosine was the typical epigenetic modification that involved in several biological processes, including gene silencing, genomic imprinting, and S chromosome inactivation. Moreover, in cancer, we can observe the abnormal hypermethylation, which is commonly found in tumor suppressor genes, and on its promoters, CpG island. Therefore, the DNA methylation is an important issue for studying several diseases and cancers. And how do we detect the DNA methylation? First, I will introduce the bisulfite. Bisulfite is a chemical compound which can be seen in this cartoon. We can see that in methylated version, which is shown in yellow circle. This yellow circle indicates the methyl group and is located on cytosine. And after bisulfite treatment, the cytosine will remain to be cytosine. However, when the DNA was the unmethylated format, we can see that this cytosine did not have the methyl yellow circle. After bisulfite treatment, it will become to the uridine. This uridine will be changed to the thymidine after PCR amplification. Therefore, we can treat DNA with bisulfite. Then, after PCR procedure, we can define which DNA is methylated or unmethylated. So, from the other cartoon, we can also see that the methyl cytosine will be treated with bisulfite. It will be remain to be the cytosine. However, when cell with the unmethylated group, it will become the uridine. And after PCR amplification, this nucleotide will become to the thymidine. Then we can perform the parallel sequencing analysis. Then we can understand the percentage of the methylated group. After that, the scientists developed a novel technology which names as the whole genome bisulfite sequencing, WGBS, which can detect the whole genome methylation at the base resolution. And we can see the methylation status in the adjacent CpG island. In this cartoon, we can find that after genomic DNA extraction, then the DNA was treated with bisulfite. Then after bisulfite conversion, we can understand which DNA is methylated or unmethylated. Then after the adaptive ligation, we can amplify the library and sequencing the whole genome DNA. This is a workflow of the whole genome bisulfite sequencing, WGBS. You can see that after sample quality control, the sample preparation, and it were treated with bisulfite. After bisulfite treatment, the cell DNA will be constructed into the library. And after sequencing the bioinformatic analysis, we can understand the DNA methylation status. From the DNA methylation, we also want to know what is the chromosome architecture. Then I will introduce two parts of this. First is that the principle of genome folding into the topological association domain, TADS, and how to detect the chromatin architecture. We can use the high-C technology, which names as high-throughput chromosome conformation capture. And the principle of genome folding into topological association domains, which means that in chromatin fibers, we can see it's around 10 nanometer, and it will contain to it will compose to the TAD topological association domain. After DNA folding, it will become about 1 micrometer compartment. This compartment will aggregate and aggregate into the chromosome territory. Since DNA is specially organized into 3-dimensional structure, so the distal DNA will be brought into proximity through the chromatin folding. This means that in chromosome territory that may contain the distant binding of the DNA structure. Therefore, scientists can use high-C technology to define the tasks, topological association domains, and chromosome compartment, chromosome territories into this figure. Then we can understand in the red square, it means the contact enrichment, means this DNA region will contact. And in a pink square, it shows the contact depletion. So we just see the red square, we can understand the chromosome architecture. And how do we define the chromosome conformation? We can see use the high-C, high-throughput chromosome conformation capture technology. It will use the chromatin dynamic by proximatic ligation. The basic protocol and workflow are shown in this cartoon. After cell were fixed with formaldehyde, disulcine-mediated glutarate. After fixation, the cells will be lysed and digest with DDE1 and DPN1 nutrition enzyme. After digestion, the DNA will be separated into small parts. Then this cross-linking the DNA will be labeled with biotin. Then after ligation, we can see the DNA will be ligase into this part. Then we can purify the DNA into the small fragment. Then we remove the excess biotin and sonicate, and then do the size selection. We can get the different fragments of DNA. Then use biotin pull down and adapter ligation. Then we will amplify the library, and we can find the final chromatin structure. Therefore, in this paper, they also want to combine the high-C and whole genome bisulfite sequencing. To combine these two technologies, they will profile the chromatin conformation and DNA methylation at the same time. Here are the small cartoons shows that after fixation, the DNA and protein will be cross-linking. Then after the nutritional digestion, it will digest into the small DNA fragment. Then after ligation, it will cause the proximal ligation. Sorry. Then after this fragment will be joined by this fragment with junction and methylation. Then this DNA will be treated with bisulfite to convert the DNA sequencing. Then using the pair-end sequencing, we can analyze which DNA is methylated and construct a library. So here is the first data of this paper. We also want to compare the contact matrix between the in situ high-C and the methyl high-C at different resolution. The upper panel shows the in situ high-C and the lower panel shows the methyl high-C. We can see that at different resolutions, their results are showing the similar results. This data was collected by mouse embryonic stem cells. And this data suggests that methyl high-C shows a contact matrix, which is highly similar to the in situ high-C. Further, they also want to compare the contact frequency distance decay curve and test topological association domains in panel C and whole genome bisulfite sequencing (WGBS). All of these three figures shows that methyl high-C and in situ high-C shows similar results, indicating that the development of methyl high-C is successful, which contains similar data to compare to the in situ high-C. Therefore, after the bisulfite sequencing, it will not affect the DNA topology. After that, they also want to observe the Pearson correlation coefficients of DNA methylation status, which were compared between the chromatin loop anchors and shuffled. This two region shows that the DNA methylation is higher in loop region than the shuffle region. And in compartment A, which is the loop anchored, also shows this in these regions, the methylation status is higher than compartment B. So from these results show that the methylated high-C can profiling the long-range chromatin interaction and DNA methyl in mouse embryonic stem cell at the same time. So they also combine these two technologies successfully. Next, they also want to ask whether this technology can can demonstrate the chromatin structure and the DNA methylation at the same time, and this technology can use in a single cell. So they also use the sorting method to separate a single cell in 96 squares. Then performing the same experiment, we can see that the after the single-cell methyl high C experiment, the bulk methyl high C and the merged methyl high C shows that the ensemble contact matrix shows similar results. These results show that the contact probability of aggregated single-cell methyl high C were comparable to the bulk dataset. Then the author want to observe whether the cell development will be affected the cell methylation status. So they use the mouse embryonic stem cell and separate into two groups. One group is the serine added with leukemia inhibitory factor LIF. And in this situation, we call the serine mouse embryonic stem cells. These cells will be characterized to be express the polypotency factors. These factors can cause the stem cells to become the polypotency. The other group is that mouse embryonic stem cell will treat with the leukemia inhibitory factor LIF, which were also added with GSK and the MAP kinase inhibitor. Then these cells will be called as 2I mouse embryonic stem cells. These cells, because the GSK3 and the MAP inhibitor 2I condition will cause the cell to stay in the ground state polypotency. Then the author compared the serine mouse embryonic stem cell and 2I mouse embryonic stem cells, their single-cell methylation status and single-cell high C status. And this data was shown in the chromosome 14. Then we can see that the contact probability of aggregated small cell methyl high C were comparable to the single cell high C dataset. Then further, they want to compare the methylation status between the serine embryonic stem cell and the 2I mouse embryonic stem cells, their methylation status. From the data, we can see that the average methylation of the CpG island in serine and 2I mouse embryonic stem cells were 61.9% and 23.4%. These results suggest that after serine stimulation, they successfully detect the methylation status is higher than the 2I status. So these data show that when we simultaneously analyze the DNA methylome and the chromatin architecture, by single-cell methyl high C, we can detect both methylation and chromosome structure in one single cell. Further, the author want to characterize the heterogeneity of cell population. Then we use the single-cell methyl high C. So the author first clustered all the cells based on the DNA methylation and of each cell. After T distributed stochastic enabling embedding T-SNP visualization of unsupervised clustering, we can see this DNA will be separated into several clusters. In serine group, we can see in red cluster, it will be clustered into the cluster 2 and cluster 3. However, when in 2I group, which shows a lower polypotency status, only show these cells are only shown in cluster 1. So this data shows that the mouse embryonic stem cells grows in syringe, the red circled, which will be divided into two subpopulations, including cluster 2 and cluster 3. Therefore, these results show that the single-cell methylation can reveal the heterogeneity of cultured mouse embryonic fibroblast embryonic stem cell. Further, they want to cluster of the aggregated methylome in subgroups, then using the tissue-specific methylome. Then we can see that the cluster 2 and cluster 3, especially in cluster 3, shows the potential embryonic linked development trend. Further, they also want to compare each cluster and the bulk in situ high-C cells, and we can see the different clusters shows the different chromatin aggregated contact. This result shows that the aggregated chromatin contact matrix in each cluster also shows a clear cluster-specific chromosome conformation. So from methyl high-C experiment, we can define the different methylation group status and the chromosome architecture in the single cell. Moreover, they also can define the different cluster of different developmental cells. So we can see the cluster-specific chromosome conformation. Then they also using the gene ontology biological process. Then they find that this cluster also shows the embryonic forelimb morphogenesis and limb morphogenesis, which are important in this biological process. Then the panel E shows that the p-value of this gene ontology biological process, and find that the differential methylation region and gene located in different compartments, which will enrich in the limb morphogenesis and appendage morphogenesis, embryonic limb morphogenesis, and limb development. This result suggests that these genes are important for the embryonic limb development. Then they also want to show some example of his experiment, how this method can be applied in our study. So they use a snapshot of the host cluster genes in differential compartment between cluster 2 and cluster 3. Then we can see that the first panel is the high-C bulk high-C in situ high-C. Then we can see the cluster 2 and cluster 3 shows the different methylation status. And these genes within the host locus 19 is differentially methylated and inhibit differential compartment between cluster 2 and cluster 3. This result suggests single-cell methyl high-C can be used to resolve cell tissue-specific chromosome architecture in heterogeneous cell population. Therefore, from this experiment, they show that single-cell methyl high-C reveal heterogeneity of cultured mouse embryonic stem cells. So from this paper, they also combine the methylation and the chromatin structure, and they can define the DNA methylome and the chromatin structure in mouse embryonic stem cells. Then they also define that this technology can be used in one single cell to detect the DNA structure and the DNA methylation at the same time. Then further, they will also find that through the different development, we can see the heterogeneity of different cells from single-cell methyl high C technology. So this technology will show will be great for the to study the embryo development. So the conclusion of this paper is that they report a molecular assay which names methyl high C, and it can capture chromatin conformation and DNA methylome in one single cell at the same time. So thank you for your attention. I'll be glad to take any question from you. Thank you.